Hi, my name is Keerthana Velmurugan, and I'm an undergraduate researcher in Dr. Anke Henning's lab, working under Sai Pavitra in the spectroscopy division. Magnetic resonance, or MR spectroscopy, is a major modality in biomedical research that can be utilized in the non-invasive quantification and imaging of the human brain. In this research, the main objective is to determine biomarkers in metabolite data obtained through MR spectroscopy of the human brain to predict what form of therapy, such as psychotherapy, ketamine medication, electroconvulsive therapy, etc., is most effective on patients with major depressive disorder. Subsequently, to achieve that, the metabolite spectra in the human brain must be quantified. So in this presentation, I'll be discussing the preliminary steps taken towards the quantification of metabolite spectra in the human brain in 2D, the obtained results, and what future steps include. First off, it is important to note that we're attempting to fit data in 2 dimensions rather than 1. The reason we do this is to clearly distinguish between metabolites. Oftentimes in MR spectroscopy, metabolite data will overlap with each other because of their similar chemical environments. This is known as J-coupling. As you can see in these plots, glycine and myo-inositol have identical chemical shifts of 3.55 ppm, causing an overlap when visualized together. This issue can be resolved by visualizing in 2D, which I will compare soon. Now back to the preliminary steps. Patient data from MR scans at 3 Tesla were already acquired, and following that, the metabolite and macromolecule spectra were pre-processed. Next, a metabolite basis set was simulated using VESPA, a Python-based software for 100 different echo times. And finally, the basis set and pre-processed metabolite and macromolecule spectra were combined into a complete 2D basis set. Over here is the pull sequence of the 3T MR scan used to acquire the patient data. At the top, we have the radiofrequency or RF pulses with a soda water suppression block, 90-degree excitation pulse, and 4 180-degree refocusing pulses. Between the last 2 refocusing pulses, the echo times was increased by 2 milliseconds to create the indirect dimension of the 2D spectra. Below, we have the gradient pulses for each dimension. And after that, the last refocusing pulse, the gradient pulses are as short as possible to implement maximum echo sampling scheme for increased sensitivity. Lastly, the acquisition is shown with the analog to digital converter block indicating the echo signal being acquired. With acquired raw data, the metabolite and macromolecule spectra was pre-processed on MATLAB. The pre-processing steps included are listed here. The reason we process the macromolecule spectra is because we have to account for the macromolecular baseline that underlines the metabolite spectra in vivo. Now let's take a look at forming the metabolite basis set. The metabolites of interest that are included in the basis set are listed here. Using VESPA, the simulation of metabolite spin systems was run 100 times with an incrementing echo time step of 2 milliseconds. The data was then constructed to form a 2D basis set. Here are the results of the metabolites GABA and myo-inositol. Here in the bright white spots are the peaks of the metabolite spectra. You can see there is more distinction between the peaks in 2D rather than in 1D. So with both the pre-processed human brain MR data and this basis set, we combine it to form a complete basis set. Here are the results from a healthy volunteer. Displayed on the top are the spectras in 1D, while the bottom is in 2D. The first column is the metabolite spectra. You can see how the peaks are distributed and very clear here. And there are some metabolites labeled on the peaks. For the macromolecules, it is not as vivid, and this is because of their lower SNR. You can see the abundance of noise in the 1D spectras as well. In conclusion, these were the preliminary steps in the quantification of metabolite spectra in the human brain. Future steps will involve quantifying the data using ProFit software and establishing biomarkers for major depressive disorder by identifying healthy ranges of the metabolites. Thank you for your time.